Okay, we're going to talk about in section 3 the regulation of mitosis. Before we start section 3, let's review a little bit for section 1. So in your groups right now, I'd like you to talk about what are the steps in the cell cycle. So in the cell cycle, we have a few steps that are really important. The first stage of the cell cycle is interphase, which the cell spends most of its time in. Interphase is broken up into 3 different phases. So right now, pause the video. What are those 3 phases that make up interphase? And what happens in them? Okay, in the first phase of interphase, you guys got it right. It's the G1 phase, and this is the cell growth phase. So the cell's actually just growing. It's also known as the gap phase. And the cell has rapid growth. It has more organelles being made. It's just everything is being replicated. And then we have the S phase. This is known as the synthesis phase. The DNA is copied in this phase. Very important that this phase goes correctly, because if DNA isn't copied correctly, well, the cell isn't going to be effective. Then the third phase is the G2 phase. During the second gap phase, the cell continues to grow and prepare for cell division. We have these hollow tubes called microtubules that are being formed, and the cytoplasm is made sure that it's doubled in size. And then we go into the process of mitosis. What are the 4 phases of mitosis? So hopefully you guys got that right. 4 phases, known as PMAT, are prophase, metaphase, anaphase, and telophase. In prophase, the major thing is that the nucleus is dissolving, exposing all of the chromosomes. In metaphase, we have the chromosomes lining up along the equator. In anaphase, the spindle fibers have connected to the chromosomes and are actually moving them towards each of the poles. And then in telophase, we see that the nuclear envelope starts to reform. Now, the last phase of the cell cycle is called cytokinesis. In cytokinesis, the cell is actually split into 2 new cells. In animal cells, the membrane just simply pinches off, forming 2 new cells. But in plant cells, we have a formation of what we call a cell plate. And that cell plate is what divides that cell in half. Now remember, we have to have a cell plate form because it has a cell wall, which is very rigid, and it can't pinch off the same way. So we have to have regulation of these cells. If we don't, we have uncontrolled cell growth and division because of protein signals and other environmental factors. So right now, before we go any further, I want you to kind of make a list of anything that you think could cause a cell to divide uncontrollably, or an environmental factor that would not allow for a cell, that would cause cells to divide irregularly or rapidly. Okay, so cell growth and division always depends on proteins. Proteins will send signals, depending on where they're at, saying, hey, a cell needs to be divided. It might be because a cell is damaged, and if a cell is damaged, we will start to see cell division occur. A protein might signal it because a cell's just wore out, and so you need to have another cell being made to replace a worn out cell. Or it might signal it because, well, teenagers, you guys are still growing, some of you, because you're actually growing in size. And because you grow in size, you're going to need to have larger organs, and so then your cells are going to produce more. Cells are continuously dividing. That's a good thing. It means that we're able to replace these cells that are damaged or just wore out. And so all the cells divide all the time. The rate at which they divide depends on the type of cell and their surface area volume ratio. Now, these signals are controlled by proteins. If a protein sends out the signal for the cell to divide, we'll start to see that happen. Sometimes you have proteins that are being sent out falsely, which means they shouldn't have been sent out at all, which can lead to some problems. So we have some environmental conditions which can cause a cell to send a protein out, or a signal protein, to start the cell cycle. Now remember, it will send out that protein if the cell is wore out or is damaged. That's a normal thing. But some environmental conditions. Toxins, nutrients, disease, these can all affect it. So let's talk about nutrients. Now, they've done studies on twins in the 1950s. Now, this is not legal, so they don't do this anymore. They took two twins and they separated them out. One twin, identical by the way, went to an upper class family, upper income, and the other one went to a lower income family. The twin that received more nutrients in the upper class family was able to produce more cells and was actually taller than the identical twin that went to a lower income family. If you are not getting the nutrients you need, your body will not signal those proteins to cause cell division. Therefore, you may not reach your potential height. So when we talk about we're much taller humans now than what we were probably 100 years ago, part of that isn't because of genetics. Part of that's because of nutrition, and nutritional standards are a little bit better now. We know that you need certain vitamins and minerals to enhance growth. And so those nutrients become really important to signal those proteins for cell division and cell growth. Another one is toxins. Toxins can destroy cells. And so if you take in a toxin such as alcohol, you're actually destroying cells in your body. And those cells then need to be replaced by new cells. And so we can see toxins can do this. You can also look at toxins as things as mutagens. These could be chemicals that could signal these proteins, which would cause uncontrolled cell growth, which could lead to a tumor. Also diseases will. Now, we've seen diseases like polio stop cells from signaling so they don't replace themselves. So people with polio would actually lose the ability to walk. We also know that if a child gets diseases at a very young age, specific diseases, it can alter how their cells divide and their cell growth. And so we need to be aware of those. And right now there's a really common one in the news. It actually doesn't affect people when they get bit with it, but when that mosquito that carries it bites a pregnant woman, it causes a slowing or slowness of the cell division within the cranial cavity of the offspring, causing it to have a smaller head. So that could be an issue too that you can see. All these things are environmental issues that will affect cell growth. Now, sometimes it signals the cell cycle to happen, but other times it stops it from occurring too. So we have some checkpoints that we have to go through. So, feedback and signals at key checkpoints in the cell cycle can delay or trigger the next phase of the cell cycle. So let's talk about the G1 checkpoint. The G1 checkpoint occurs obviously before the S cycle or the S phase, before DNA is replicated. And it makes sure the conditions are favorable for the cell's health and to ensure that the cell can grow large enough. If these conditions are met, then the cell will enter into the synthesis phase or the replication of DNA. If conditions are not favorable, the cell can go into kind of what we call a resting period, which means it will stay within this phase, this G1 phase, for an undetermined amount of time. We do have certain cells, such as our nerve cells and muscle cells, that will remain in these resting periods for long periods of time, and they don't divide as often. And so we do have other cells that will go through these much quicker, like our skin cells. And then we have our G2 checkpoint. Our G2 checkpoint happens right before mitosis occurs. So right before prophase, metaphase, anaphase, and telophase starts. It's checking for any mistakes that may have happened in the S phase. This is important because if there's mistakes in the DNA, it is going to affect how the cell works. And so basically it's the protein. It's also double checking to make sure the cell is also large enough so that it can divide into 2 daughter cells that are equal size. It's making sure it has all the organelles needed, and it's really just looking for everything there so that we can go into mitosis. And if it's there and everything is met, then it releases a protein that will trigger mitosis. If it's not met, this can end that cell phase, or it can stay in there for a delayed period of time. Then we have mitosis checkpoints. During metaphase, where the chromosomes are lining up on the equator, this is a checkpoint to make sure that our chromosomes are going to attach to the spindle fibers and divide equally. We do not want to see one cell with more chromosomes than the other. So without this point, the sister chromatids for one or more chromosomes may not separate properly, therefore causing a genetic mutation within the cell. Now, this could be harmful to the cell, or it could be helpful. Most of the times it will terminate this. However, some points we have some cells that are going to go unchecked, and that's when we have some problems to occur. One, they can develop into a growth of uncontrolled cells, which we know of as a tumor, and is going to be in the second part of this video.